introduce Cong, who I got to know as a PhD student when he was about to publish the first study on CRISPR utilized in human cells. So that was a huge landmark, of course. And then I got to know him better later on when we both did our postdocs in the Viva lab. And it's no surprise, I guess, that since all that pioneering work that he's done, that he's continued to do so in his own lab at Stanford University. And he's today giving a talk about how they develop machine learning optimized CRISPR tools in a melanoma model to look at drug resistance and immune evasion. He's had too many awards and honors to go through the list, but for instance, HHMI International Fellow Award, the Genomic Innovator Award, the Donald and Delia Baxter Foundation Faculty Scholar, and so I won't go through the whole list, but it's such a pleasure to have you here today to brainstorm, as always, and look forward to your talk.
Thank you. It's a great pleasure to be here. Thank you, everyone. Really love to just to be as interactive as possible because I'm planning to present all three aspects of the work from really more tool development to cancer application with collaborators and also all the way to kind of very computational work. So really hope to share with these three, hopefully aspect of the lab's research. And I think all of them kind of connected to, as you mentioned, technology development. That's been how I first met you're in the Beef lab, and so this goes. Sorry, started very quickly. This is a group. We were mostly students and postdocs that are working at the interface of communication and experimental biology. So I wanted to quickly go over three, I think, really interesting technology-driven work that we've done. First, how do we do, kilo-based scale, large-scale gene editing using new molecular tools? And then second is really thinking about, in addition to just developing the perturbation, how we can use these perturbations to study this CRISPR gene editing tool to study cancer immune interactions. Finally, as we're getting more data from these gene editing experiments, single cell experiments, we have been also thinking about ways to leverage AI machine learning in help us analyze the data coming from these experiments. So first part, maybe focus more on the tool development side of the lab. I wanted to share a bit about how we have been thinking about pushing the limit of genome engineering, genome sort of insertion. It's a better way to perturb genome function. And all of you are very familiar with the kind of CRISPR systems. And we have been particularly interested in, we think, one of the most challenging tasks in a human cell, which is large-scale kilo-based genome integration insertion. Traditionally, it's very tough because when you cut a piece of DNA, the dominating event that happens in the lab is just break the DNA up, random mutations, which is really great for knockout gene. But if we want to integrate a large piece of DNA, like do a protein tagging, do a large-scale gene correction, usually it's a less preferred pathway, traditionally done by, for example, homology recombination, where we have a donor template, and through a complex repair mechanism, it replaces the original sequence. But usually less preferred, right? It's more work. So we have been thinking about what are the ability to push that part of precision replacement insertion more efficient. And very quickly, there are a lot of interesting tools available. Transposes and SAMS server like here has been pioneering that as a tool development. Those were prime editing with recombinase. Also like double prime editing with recombinase. A lot of really cool tools have been developed. We took a little bit of a different approach. We look into more coming from what's most simple for nature in terms of doing gene insertion. And one of the simplest systems we found in large genome insertion is coming from also, not surprisingly, from the simplest organism that needs to do this, which is bacterial phage or virus, right? That's bacteria. So phage, the yellow part, we can imagine this is a prophage, right? Like the phage genome. The best way for phage to, without killing the bacteria, replicate very, very efficiently is to integrate itself into the bacterial genome. And they have a very elegant two-protein approach to do this. Exo beta, or sometimes also called recombination. The idea is that you have an S nuclease that exposes homologous sequence at the end of the phage genome. And then the phage genome have evolved what we call almost like a recombination hotspot or landing site that resembles bacterial genome, or here it's target DNA. And a very small recombination protein called beta is specifically binds to single-stranded DNA and mediate a homology exchange to integrate, for example, a phage genome into the bacteria. And this is what we call recombination or recombination. And the core protein beta is what we are really interested in because it's one of the only family, if you think about it, most recombinases are double-stranded separate, right? Cree recombinases, large P, right? These are very efficient, but double-stranded DNA. Beta, or something called single-stranded DNA protein, SSAP, is the only known major family of proteins that are single-threaded recombinases. So they operate on single-threaded DNA. And that's why they're super compact, but also very efficient. And interestingly, very recently, from work by Luciano Marfini's lab in Rockefeller, they figured out that the phage actually leverages to counter CRISPR cutting. So we have CRISPR cleaning the phage genome. If some genome got cut or got cleaved, but some other phage hasn't yet been cut by the CRISPR, the phage can recombine through this iso-beta mechanism shown here to repair itself, to avoid being cleaved by CRISPR. So this is almost like a natural anti-CRISPR system. So we were really interested instead of using it in bacteria phage, can we leverage this system to program or to insert a large piece of DNA into human genome? So what we did is instead of cutting the DNA, we're hoping to have a natural way to recruit these proteins. We use a dPAS. In this case, it's really binding to the target, and then we can recruit the SSAB protein. We have various ways to bring it. One is to use afterward, but to make it very brief. Once you can recruit SSAB for a target genome through Cas9 binder, you can start to see very effective docking of the donor DNA through the single-stranded exchange mechanism. So we wanted to, in some way, make the system resembles a RNA programmable pre-recombinase, where we can just change where the Cas binds to the genome, recruiting the SSAB, and then the donor will be updated. We have screened over 600 different SSAPs, and very much like CRISPR, the phage have evolved a huge diversity of these recombinases. And we're working with Alex Cong in Joseph Boninomi's lab at CSF, which is a very excellent short collaborator, very known for their phage biology work. We screened many of these proteins and found some of the SAP have really strong activity in human cells. And this is because these are about to be pre-printed, so I want to be keeping a little brief. But briefly, we can now deliver these proteins because they work on single-stranded DNA. A natural way to deliver them is to use AAV, because AAV, as most of you know, are delivered as a DNA format. It's a DNA virus. So we initially tested using a double AAV approach, one AAV encoding the Cas and the recombinase, the other one encoding the guider and the donor into the mice, and you can see pretty efficient knock-in into the liver, which is the natural sort of target of these AAV8 particles. And more interestingly, of course, more relevant maybe to the cancer application, we have been working with Jackson Adams at UCSF to start screening for what SSAB candidate might be more optimal in primary human T cells. So this is still ongoing work, but initially we tested on beta-2 microtubulin, one of the a very good target for potential cell therapy. This has been used by Justin's group for engineering CAR T cells. So we take primary T cells, expand them, and then do the editing, and then later, in this case, initially we want to kind of simple manner, so it's knock-in a GFP cassette into the beta to B2 locus, and you can see across the group, if you don't have AAV, you just have this Cas9 with AAV, it really has nothing. But once you start with SSAT, well, the increasing amount, depending on which specific kind you use, very good annotation. Sorry, things a little bit for the annotation. So these are naturally single versus double knock-ins because we can see different strengths of GFP knock-in depending on whether it's single or double. So we can see that we have to 70% of the cells total that have effective knock-in at B2M. And we're now, and this is roughly, 1 kilobase of cassette with the GFP in the poly-A channel. So we're now testing inserting a whole CAR cassette into this locus, and we're also testing additional locus such as which is the TRIC locus, that's some additional locus to see whether we can robustly targeting multiple locus in T cells. But in general, the general sort of approach seems to be working both for mouse liver and also for primary human T cell. So we're really excited to start applying these tools more broadly for example, knock-in, cell therapy, cell engineering. But also we're very interested in thinking about maybe if we can get this efficiency really less targets, we can start doing some large scale sort of knock-ins using this approach. And the benefit of this approach is that because it's usually single stranded DNA as a substrate, we observe that there's very minimal toxicity. We apply them to primary T cell in vivo. And also because we're using not a double-stranded break mediated mechanisms, we haven't observed substantial concerns of off-target translocation when we're using this approach. But definitely more tests are needed. So hope to share more extensive test results very soon. And one ongoing small piece of data I want to show, this is still very early, is that we also wonder if you think about Prime, which is a very powerful tool. Prime editing actually goes through a single strand intermediate. So I don't have a theory, if you think about it, Prime editing basically use R to extend a piece of DNA and hope that that flag of extension from Prime editing will be integrated into the genome. So we are starting to test if we add SSAP to a Prime editing system, would it enhance, basically the editing efficiency of Prime editing. So this is the initial work where we're using a simple setup that sent to actually this was back when I was as the other in doing my postdoc in the VIV, I met a friend Francisco who was actually a neighbor who now had his own group at MIT. So we got a recorder system from Francisco and we tested using PEMAX and also PEMAX plus the different SSAPs with screening. And we can see a substantial improvements this splicing receptor correction is used by Francisco's lab in their recent paper. So we think it seems to be promising that by stabilizing and improving the single-strand flat integration, maybe the SSAP protein can also enhance Prime editing efficiency. In more preliminary tests, but of course, hoping to test it across more locus and more types of edits that were making to the genome. So broadly speaking, we think that we have a single-stranded DNA intermediate, or we wanted to leverage single-stranded DNA donor in the case of A and B, or we also have some data on circular single-stranded DNA, which is very easy to deliver because it's also less toxic. I think these are very beneficial in terms of using the single-stranded recombinase. And maybe very interestingly, recently, Dr. Bell's group and Poland group, two groups who studied these proteins, saw one of the first crowd-EM structure of SSAP, and you can see that it works as a kind of complex, and you can see the two-strand of DNA and exchange, almost like within sort of the cleft or the sort of the channel that are formed by the SSAP protein. So this is why we think that it requires a really strong homology in order for the system to work, because it really is kind of this narrow channel that anneals DNA together and force a DNA exchange reaction that helps with recombination. So with that, I wanted to start moving on to the next part of my talk, where as we're designing these tools, we also wanted to apply genetic perturbations, really understand mechanistic sort of aspect of cancer immunology and cancer biology. So in the process of this work, I wanted to share some of the recent work on leveraging CRISPR systems for perturbing and tracking cells, both in vitro and in tumor immune sort of interaction. So primarily our motivation was to think about how do we track cells as they evolve. And this work was started when we have an initial discussion with Marty Winslow, who's a cancer biologist at Stanford, and the project of Marty's lab is really to understand cell evolution that are sometimes called cancer stem cells, but also is also a key step in terms of thinking about drug resistance and metastasis of cancer. So initially, one of that work is myeloma and lung cancer. Starting with myeloma, given the very good model systems that we have in the lab with my team, we know that we'll have myeloma. Some of the very effective drugs are BRAF inhibitors, such as PLX, whereas these drugs initially need to be very fast because of the minimal residual disease that are often associated with this treatment, the cancer come back. So you can see that after about 6 months, even though the initial response was very, very good at 3 weeks time point, 3 months time point, after 6 months, the short myeloma came back. And what we are thinking about is, can we track cells as they develop this resistance, and then as a way for us to identify new mechanisms of resistance? So as a potential new drug. So to track these tumor lineages, what we decided to work on is CRISPR-based tracking. This figure hopefully show you the motivation for using CRISPR as a barcode tool for cancer evolution. So typically, like whether it's a fluorescent protein or maybe like a genetic barcode, what typically happens is you can initially track the cells, and then eventually all the progenies will have this label and the fluorescent protein genetic barcode at time point 0. So you really get clonal population at the end, but you cannot tell the difference between, say, this cell versus the other daughters. But with CRISPR system, because the editing happens over time, imagine these are 5 different CRISPR edit sites. And when CRISPR edits this kind of stochastic process, in this case, then you really have a lot of edits that happens over time. And by the order of the different edits happens, even if you just collect cells at the end, you really can understand, for example, these 2 cells are actually siblings, right? Because they share 2 out of 3 edits. And these cells share another 2. So we know that these are more remote sort of connected, but these 2 are really close siblings. So you start to really be able to see the family tree of all the cells, to be able to think about when, for example, the resistance happens, what are the driving forces of expansion of the cells, even if, for example, there's some genetic changes in the middle of the resistance process. So we think that using this CRISPR barcode, you can have a really full reconstruction of the lineage over time. And this is even diagram here. Again, you can see that different edited will have different mutations. So and so even if the edit happens at similar target sites, the stochastic nature of the edits, you really can start labeling a lot of cells because of the edits in general are very randomized and stochastic. And all is coupled basically perturbation with construction lineage. Then of course both of these information are DNA coded and read out through single cell RNC or single cell omics. And I want to mention that in addition to our approach, there are also a lot of interest in the field to develop similar approaches. There's a lot of requests that are using stem cell tracking, and then there's also from Jason Urelax a primitive-based barcode. Those are all very similar tools. And our work is more focused on Cas9 because we wanted to leverage, as we'll see later, the multi-class editing capacity of Cas9. Maybe to give you a little more information, like small system, there are a lot of Cas9 based barcodes, but Cas9 basically for every single CRISPR RNA expressed with Cas9, it's added a single site. So that's how you can see there's almost this literal gene how large your particle gets, how much you can track cells. Because every single CRISPR construct can add a single gene. So there's kind of this scaling inside it. Cas12 is very exciting to us because it actually is a dual RNA mix. So Cas12 protein first process a free RNA into multiple single RNA. And so that even if you have a single free RNA, you will target many different locations, up to 20 that we have tested. So the idea is that if you look at here, what we do is we can have an array, basically a single construct that can process by Cas12 into multiple guides and add multiple targets. And this way, you can see that we can add, in this case, 4 different genes, so 4 different locations within the same gene, or we can add 2 genes, each with 2 sort of guide RNAs. So we can also add 4 genes, right, within a single process. So it's a very scalable system. And there's no change in the construct theory. Like, always use a single construct to target as many genes as you want. So we do think that this is very attractive if you wanted to add multiple locations and also to determine the cell at the same time. But one problem we ran into early on is that how do we design the sequence if you can target any sites? So very briefly, we actually use a machine learning in silico optimization approach, where we use the AI to model sequence diversity and the efficiency of Cas12, and then basically very quickly improve the editing efficiency of these Cas12 constructs. And so after doing optimization system, because it's published, I'll go maybe a little more high in each part. We can now have a very small constructs that track almost the same sort of number of cells at the same pharmacokinetics as some of the really large constructs that people have used using Cas12, thanks to the multi-target editing of Cas12. And so we now started with pilot systems. This case for visual or left deliver the barcode, and then we can introduce the barcode editing over time. And then over different time points, we can basically have collection of cells, reconstruct the barcode lineage history as well as gene expression at single cell level. And this is an example of a melanoma resistance pattern at day 7, day 14. You can easily see that some of the resistance cohorts are starting to extend over time. And this is using PLS, so BRAF inhibitor treatment. And then we can also not only have the clonality, but for each of the clone, we can also reconstruct every single cell's history. It almost like their family history through this CRISPR paradigm. And one thing that was found very interesting is that if you look at what are the most enriched genes that drives subclonal expression. So within this clone, for example, the most example, what are the signatures that drives subclonal expansion? We found very interesting set of genes that in the PFC2 complex that are enriched in these, high memory. Memory means that it's always enriched in the same cell lineage in the cell. And then we also found, for example, all these genes are enriched in EZH2 chipset data, which means that EZH2 is indeed binding to this every gene site. And when we tested, it use different treatment. This is a single BRAF inhibitor. This is a single MEK inhibitor. This is a combination. So this is a single EZH2 inhibitor, and if you use a double inhibition of both BRAF and EZH2, we have substantial improvements of killing the resistant cells. And also, if you look at TCGA in melanoma patients, the combo of these 2 genes, meaning that if you look at EZH2, low expression with high expression, these actual high patients have a really substantial lower select, meaning that a combination of EZH2 inhibition and BRAF inhibitor will be really potentially powerful in melanoma treatment. So if you look at clinical trial that's ongoing, there's actually a recent wave of clinical trials that are in phase 1 and I think mostly in phase 1 now, that are using combination of BRAF inhibitor and index2 inhibitor for basically melanoma, which are very, very sort of correlated with what we are fighting with. So we're doing that by looking at the dynamic sort of history of these cells. We can start to understand resistance programs within these evolving sort of resistant population that are otherwise very difficult to study. And then more recently, with Will Gridley's lab, John Sang-Woo's lab at Stanford, will be pushing this further to what I described as both doing the barcoding of cell lineage and also the perturbation of cells, where the Cas12 really provide a very powerful system for that. And here the setup is more complex than what actually already in the moment, where we're culturing basically myeloma cells with NK, primary human NK cells. And as NK basically are killing the tumor cells, we start redamping. The initial sort of NK addition, the first round of NK killing, and then the second round of NK killing after sort of the cell goes through the first one. So we really are trying to understand how the NK killing process are evolving with tumor cells using the single cell approach. And this is a expression changes. We see very substantial difference between the pre-NK treatment cells versus the two post first one, second of NK killing. But more interestingly, because we are working with Gridley's lab, we also collected the chromatin accessibility data from the taxi, and you can see a very substantial shift of a taxi peak upon the NK treatment. And then when we now start looking at the modality, these are two clones where we tracked the most abundant sort of signals are recovering. You can see a very, with the lineage recording part, you can really understand the dynamics within the clone. Can basically observe very interesting expansion of some of the clones over time, and also essentially very interesting dynamics. For example, like expanded the initial treatment, but then also seem to lose their ability to resist NK killing over time. So this gives us start to have this really fine resolution to understand within a population of tumor cells how each individual dynamic response to NK killing can be regulated by looking at, for example, compare these two clones that opposite dynamics, what are the genes that drives, for example, this evolution process. And so here, we're starting to see some very interesting genes that are showing up as resistance clones that are upregulated from the resistance clone versus non-resistance clone. And some of them are metabolic genes, some of them are genes that are very poorly studied in the past, implicated from the patient serum data in, for example, multiple type of cancer. So we're starting to really dive into this individual gene that we think drive the difference between the fate of the resistant phenotype within the NK killing process. And for the depleted population, which are being easily killed by the NK, we find an enrichment of TGF-beta induced pathways, which is interesting. So the idea that why TGF-beta inhibitor may not be a great cancer drug is that perhaps while you are people have proposed TGF-beta as an interesting pathway, and some people may know that in T cell biology, TGF-beta is a very substantial sort of this idea that it is going to facilitate exhaustion. So if you give TGF-beta inhibition, you will activate the T cell hopefully. But then in organism, we're seeing that if you just broadly inhibit TGF-beta, you might actually reduce NK killing in this case, so that you might have this counteract effect from broadly inhibiting TGF-beta. So perhaps the better way to do it either to specific targeting T cell, right, as a more targeted engineering approach versus just using broadly TGF-beta inhibitor. But these are really, I think, initial analysis. But now we're hopefully diving deeper into sort of the validation of individual genes. Here, this is an example where we're basically starting to engineer NK cells in the target formation in vivo This is one of the first experiments we did, where we're targeting actually one of the genes that showed up here. As I mentioned, we're actually initially testing, CD55, and then you can see that in CD55 pathway, you can really achieve a substantial difference. This is looking at a model NK cell line, not primary NK or NK92 cell. This is the NK cell line, but it's also humanly right. So we can see a dramatic difference in the tumor size over the initial test we did. But now we're doing this for more of the time in vivo to really understand if these interesting genes we discovered from linear tracking perturbation are really going to have an effect in vivo. Then that was tool that we developed more recently. So this has just been published. Is a mice model, which allows us to do all this previous work instead of in vitro but in vivo. And you can see that supported by the U54 program, we made a Cas12 model, Cas12 mice model, where all the cells have a pre-dependent allele, where essentially it's create lines to drive the expression of Cas12A, which is the core genetic protein. And you can see very nicely, so if you have pre-activated, you have broad expression. Well, this is using a CAG create. So the CAG promoter will drive pre-expression in all tissues. But imagine you have for example, a T cell specific pre-driver line that can drive only expression of T cells. We have now been able to complete validation of this model. We also are now seeing very good barcoding over time as those perturbations. This is actually a sequencing result showing you the accumulation of edits over time. So you can see everything accumulating over time. So we really are starting to see that using this mouse model, we can do a lot of in vivo perturbation tracking using these Cas12 mice. So very exciting to now starting to share this mice. So this has been deposited with Jackson Labs. So everybody can collaborate. And I would say that this will really give people ability to do in vivo perturbation tracking using these Cas12 systems. And finally, I want to, we have more data than I'd love to show for the Cas12 people interested. But in the final, 10, 15 minutes, I want to also share about what we have been doing building computational to hopefully make the analysis of all these genetic systems and design these genetic experiments more straightforward. And so, a lot of people have been using AI in their work, but as this is an example from OpenAI's text, right? In different subject matters. You can see that in chemistry and physics, all these recent, AI models for a lot. But the technology, there's very little improvements in the type of, biological aspect questions that the AI can answer. So what we have been thinking is we needed to have very specialized AI models and systems that are made for biology, for biologists, that can really help us in real experiments. So the framework we have been thinking is called ABC. So maybe the A is for agents, where you can really have a team of AI systems to help you do tests. But this system will build out not just the ChatGPT, right? But it's various models that really understand biology, like protein language models, like AlphaFold, other models, and different types of single cell genomics models that are very much built for biologists, but not for general purpose. And finally, we also see that in order for us to understand if the AI is working, we need standard benchmarks that help evaluate the systems. But this is what we call like an ABC approach to think about how we use AI. And one of the things I want to share is we've been focusing on building RNA formation models because there are already some really good protein models, as some of you may know, AlphaFold, ESM. There are also DNA models like EVO2. But RNA as a very useful molecule in biology, there hasn't been a lot of recent work in RNA formation work just showing up. So we started to build various RNA formation models in the hope that they can start understanding RNA sequences and even design sequences. So this is published last year, where we already are starting to build models for mRNA because we publish, I won't, work, spend too much time. But this is a work I contributed this year, and it's now under review. But this is our first work where we take sequences we can find from various public databases, and we try to build a generalized RNA model where it doesn't just understand RNA, but also understand non-coding RNA, small RNA, all various type of RNA, and taking data from the database called RNA Central. There's also actual data that we collect ourselves, and we can feed this data into a transformer-based architecture, and then connect it to a diffusion model for generating sequences, similar to what diffusion model have been able to generate images or protein sequences. So the idea that by taking large amount of sequence and then encode them in a transformer, and then be able to use diffusion to design sequence, we can perform various different tasks for RNA sequence. And here's the example. We can predict our structure, and prediction from this model is actually even better than some of the most sort of public available tools. Shown here on the public, you can see this is the experimental structure. This is from the most recent RNA flow. And this is from Genesis, which is our model. You can see that our model prediction of the second structure closely match the experimental structure much better than, for example, the RNA fold prediction. So it seems that using these large models, we can really predict RNA structure very reliably across different RNA. And we can also start to design different, molecules. So the first thing we experimentally validated is to design the CRISPR RNA, right? This is the RNA that we've been using a lot for our own work. So we started with, the most common used SB-Cas9. There's a 78 base pair scaffold that everybody uses, right? Which is very complete. But it's natural to that. It wasn't evolved, for example, in human cells. So we leveraged our genesis to design a panel of different RNAs and experimentally screen the 16 different RNAs for which one, 24 of them, to see which one really is the best. Interestingly, if we test the top candidates for base editing and prime editing, which can definitely improve, particularly for CD, skeleton base editing, and also for prime editing, very substantially over the natural, CRISPR guide scaffold used by most of them, right, for SB-Cas9. So what we think is the idea that instead of evolving in human cells experimentally, you can feed a target RNA into the AI model and instead evolve the RNA to get hopefully a better performing sequence in human cells without doing any experiments. So we're now testing the base editor prime editor scaffold across many different parties with collaborators. And very interestingly, the order is a little bit different. But if you look at the hydrogen binding between Cas9 protein and the design RNA, you can see this is the hydrogen bound length of the native original one-time CRISPR Cas9 from bacteria. This is our best candidate scaffold using AI design. You can see that shorter hydrogen bound, meaning that even from a physical perspective, it's making sense that this AI design scaffold has a shorter band length, thus a higher affinity for the Cas9 protein in human cells. And this way we can explain what we're seeing in better base editing prime editing using the newly generated scaffold. So this is hopefully show example where if you take a huge amount of sequence data and try to use machine learning to embed it into a larger model, you can start to do in silico evolution of sequences that are very useful potentially for downstream tasks, in this case, gene editing. So we're really excited to potentially use these to enhance what I showed you earlier in terms of perturbing cells, in terms of tracking cells. So really, I think about this as a very efficient way of evolve RNA sequence design. And so the final part, I want to show you some recent work we have done building up these biology foundation models to help maybe everyone to do more complex tasks, even AI agents. And so AI agents basically is, you can think about it as just a virtual team of AI that will help you to do different tasks. In this case, this is the first agent we built on CRISPR GPT. It's basically an AI agent for gene cell engineering. But you can see there are different agents that does different things. So, there's an agent that specifically plans for the experiment. And then you can see this part is the various agent that is doing the experiment design. Then you have an agent that interacts with the user in a natural language. There's also agents that are calling specific tools. So you can see it's actually almost like in the lab manager, right? You have also, all the trainees, the students that are doing the experiment, and then you also have, for example, a core facility that provides you with tools. So in some sense, AI agent is just a virtual team, right, to help us with different tasks. And to evaluate if these AI agents are really helpful, we're going to what we call genetic investment, which covers things like experimental planning, design, delivery system selection, troubleshooting experiments, and some set of things that are usually come up when you do a CRISPR DNA experiment. And we can see that using the CRISPR GPT system, a lot of the work that we typically can be done can be solved almost to 90% of the time using these AI agents. Maybe to give you a more specific idea of how you use it, we're going to hopefully by the month release this website to everyone. In this website, you can maybe I'll pause here. So you can basically create projects, almost like similarly using benches with people. And once you create a project, the cool thing is the AI will have different mode. One mode is if you step-by-step predefine structure of the task. Like I want to just design CRISPR RNA, then it's like this one. But what I'm going to show in this demo is the auto, where you can just tell the AI anything you want to do. And it will basically generate the workflow on the fly for you. So in this case, you can just type in natural language, so for example, I do this experiment, and then the AI will start generating that you can see in real time, where, for example, in this case, this is performing loss of functionality in the lung cancer cell line. It will give you the different steps that you need on target optimization, optimization, prediction, all the things that you need automatically. And then it will give you suggestions, and you can basically make corrections if you want it. But essentially, you don't really have to perform most of the tasks yourself. It's really interacting in natural language in AI. And it will give you all the different pieces of information you might need. And the cool thing is this AI has actually been learning all the experiments, not only from the public database that we have already trained into the model, but also your own lab. Own experiments. So the idea that if you are doing all your CRISPR design in this centralized project sort of interface, your previous experiment will help you to understand what worked, in your previous maybe lung cancer experiment, what didn't work, and then you can iterate, providing better and better hopefully performance for experiments. And also we made sure that everything in the AI agent is clickable. Like for example, if you wanted to find a paper, you can literally just click the link. It will take you to a real paper. And then the nice thing about AI agents is that, for example, if you wanted to analyze the data, in this case, I'm trying to analyze a set of NGS data, it will automatically understand the content of the experiment. So you don't really have to write any code. You can just upload your fast-field sequencing files, similar to if you're doing cluster, and then you will be analyzing the data in the background and sending the results. So once everything's done, it will generate a report for this whole experiment, showing you everything you've interacted with, every sequence of design. In this case, for example, this is the guideline you're using. So it will have a link to all the output from the computation analysis that was done in the background. So the idea of using these agents is, I think, twofold, right? One, if you have a new student now coming into my lab, they don't really need to have a senior PhD or postdoc to work with me, at least not for the type of work that we already identify or integrate into the system. They can just interact with the CRISPR GPT to design everything. And we also have the track of everything the students or the new postdocs doing to go back and look at it. And also all the experience, all the data can contribute to your lab pool of knowledge, continue to help everyone who's coming to the lab so that you can capture, for example, knowledge experiments automatically using the AI. And one thing that is in the paper, sorry, this paper that was published last month, is that we, in the prosecuting agent, would take about 11 years of genetic knowledge by top scientists, including our own labs for sure, but now with Sandanda's group, for example, from North Genome Center, from Patrick, a lot of people who contributed to the data set of their own sort of labs knowledge, for example, helping to improve the model beyond, for example, a single last knowledge, which is what I think will be really cool. You can do this not just for CRISPR, but for example, single cell analysis and for other things. And as I mentioned in our lab, new students coming in are really starting to use these tools to drive real experiments. In this case, lung cancer. And interestingly, for example, recently we have a climber who come to us want to do a genetic work in Alzheimer's disease. And we didn't need to send a student to his lab. We just had their lab using CRISPR to design everything. And then it actually worked well for their work, and the paper has been published already. And finally, as I mentioned, we're working now on expanding this CRISPR agent to broader use cases. We are running frequented agencies called Leak, which can perform various genomic tasks like single cell analysis, data analysis. Hopefully, by the end of the month, we'll also put a new work called auto screening, which we specifically designed for various types of functional genomic studies, where you can basically have AI agents do deep research and give you examples, give you sort of automated analysis of CRISPR screens, RNA-seq analysis, GWAS study. More specifically, take CRISPR screen as an example. If you have a specific question, what humans do would search literature, would check previous research data, would check pathways and different databases, and then we can design library. If we're not running genome experiment, or if you run already have screening, you do the same thing when you look at the top hits from your screening. So these are all very much done by human. But what if you have an AI agent, right, that to literally for you based on your request or based on your data, and then it will select various different aspects of information that's useful, gene ontology, previous RNA-seq data, virus screen data in the literature, gene expression of disease database like TCGA, cancer, patient sequence data, all the different things. Do an exhaustive deep research, and then reviewing and summarizing all the search results automatically for you to basically help you as a research assistant. And so, to show you an example, if you have already done a CRISPR screen, but another screen agent, it will generate a series of table for you where we list all the genes that the agent is searching with very different aspects. Why the sort of the database or data source is very relevant. Also, you can actually click with them to see specific analysis, like kind of pathway, different disease association. So everything is clickable, so that you can really understand what the AI did and how it is ranking. And you can see that there is a score where the AI is weighing different sources of knowledge and give you a priority of which genes are very interesting to study in your case, or which, if you already did a screen, it will give you the re-ranking of a screen database. And what we did is we tested the agent on 2,000 CRISPR across 384 applications from the Valkyrie database. And you can see that if you use I think that's the basically blue line is using our agent. And the red is actually using other agents that people have published in journals. And the blue line at the bottom is actually using ChatGPT. You're going to see that when you the screen agent we develop, you can basically have 8-fold less gene than you need to screen. Or if you screen the same amount of hits, you will get 4-fold more hits using the AI tool, based on the data from these 2,000 risk estimates. And what we also did with a lot of the screening is that if we take available screen that published, compare the predictions from the AI system and the experimental groups, which is usually in the raw data, we can then do reinforcement learning to teach the AI how to understand different various type of evidence for the screening. So with that, we train our own sort of model that specifically are really good at doing deep research for functional screening. And this is what you can see, if you, for example, so we tune our own model versus the baseline. We're going to see a substantial improvements over the baseline model for functional studies. And then we actually published a tumor screen data, as mentioned, what we do in the lab, where we basically compare NKT the resulted group in a genome-wide CRISPR screen, and then we feed the screen data into the AI agents, and then be able to ask it to rank, to re-rank the top hits from the original. You can see that, for example, this is the example of the 3 genes that are coming out from screen, which are very interesting because these genes have the highest chance of ranking after you use AI. So, for example, CNAM1, which is known gene in NK biology, rank consistently high. So, in a positive control, the AI agent understand it is still a very valuable sort of gene. But CNAM5 and CNAM6, originally in the raw data, we're using MAGIC, for example, the raw data, they rank, 155 or 143, very low ranking, relatively speaking. We wouldn't probably have picked it for validation. But after the auto screen re-ranking it, all the rankings of these genes substantially improved based on the different literature search and data analysis that the agent did. And so we did a individual gene validation where we targeted A375 cells using CCAM6 overexpression, and we can see a substantial improved relative cell fitness, or basically, you basically see a dramatic effect in the fertility for CCAM6. And as you know, CCAM5, CCAM6 have been already been, especially CCAM5, have been already been used in phase 1 studies where people are targeting CCAM5 using ADC as a natural tumor therapy. But in our case, we think that CCAM6 is probably also very targeted based on our reanalyzed CRISPR screen. So I think this is an example where whether you are first designing a screen or you are trying to find targets to prioritize from existing screen, using this deep research agent, it can really help you prioritize genes for functional genomics. And the other example we did is we have a collective that are trying to understand cell network fusion events for heart disease. And in using the agents, we basically redesigned the library. And a typical process where a human takes 3 days, in 2 minutes, the offspring can complete the whole design for the scientist. And then if you perform the screening and also the individual validation, we found very good results from targeting the auto-screen design genes that are very effective in terms of identifying genes that are really having an effect in the actual experimental setup. So in many ways, we think AI agents can help you both design the screen better, but also analyze the screen better. Now, most importantly, it saves a lot of time so that you don't have to read the paper, you don't have to do the data analysis for a functional genomics study, but you can just basically let AI run for you. And one thing that I was discussing with Jan earlier is that we have also been expanding this to multi-target screening, where we can do combination, design for libraries. So this is still ongoing, but what we're really excited about is to, think about we'll have a combinatorial targets library. The space is huge, Or if you say targeting genetic screening with a drug treatment, right? There's a huge space of design that you have to navigate. I think this type of agent, agentic approach will be most helpful. The design space really big and really needed, hopefully, a very, helpful way for AI to help you at least narrow down the targets, and then you can do human review. So we do expect that for CRISPR screens, for infectious expression, and for GWAS study, we can really use an AI-driven deep research approach to give a very nice ability to review designs, generate designs, or even to reanalyze and provide screens. And what we're trying to do now is to reanalyze all the different, about 500 different, target discovery screens, over 12,000 applications, to really give everybody sort of this resource where you can, essentially take all this data automatically when you're doing your own screen design and screen analysis, through these AI agents. And this really has been driven by really our own work where we don't want to have, right, like someone sitting in the computer for days, reading all the papers. Ideally, you'll have AI systems that you can trust that embedded in the web that can help you do a lot of work. So with that, I'm going to conclude my talk here today, where I really think that we came a long way as a group where we started more with developing genetic perturbation tools to the tools with parameters to study cancer evolution, and now more recently tumor immune interaction, and then finally, more recently we've been moving more towards, once again, all the data from these function generator studies, how do we use AI and their systems to analyze. So this three sort of connected story gives you a sense of how the group has been trying to move from all three different aspects to provide more powerful tools for function generator. Thank you very much.
Great. Let's open it up for questions.
Sorry. Yeah. Thanks for the fascinating work. I just had a question about the AI related work. Obviously, there are things that are really, really hard or impossible for human to do. Like if you don't need to design, protein sequence to forward into such a structure, I cannot do it. But also it kind of bleeds into things that human can do, but might just require a lot of learning over time, right? And obviously the benefit is like productivity and time saved. I was just curious because you were saying that you already have implemented it in like so. Is there anything that is lost, you think? Just curious about your take on that.
Yeah, I think that this is a great question. Thank you. We found that I think two thoughts here. One, for example, for a lot of the sequence example, like the sequence vision set is very hard for them to do. So having kind of the model and specialized models and also the agent to inconveniently call these tools and models to do tasks for us has been extremely powerful. Which is why we're still exploring, and there more work needed is on the tasks where like probably human can do it. And it's not necessarily a simple like yes or no answer, like the auto screen example. So we can probably still make mistakes, whether it's human or AI, thinking about analysis, and also for single cell, right? Different people may have different ways to analyze the single cell data. So for this type of task, we think that there's still a lot more question mark now. Whether we really should trust everything that the AI generated out, or it's more like AI give you perhaps we found something interesting really that I think the AI can give you at least a list of options. So you don't want AI to say, okay, give me 2 genes that that are the most exciting something. So maybe make it a little more like a filter, if I may call it, of hypotheses or of interesting possibilities using it, which is more efficient to things while you're sleeping or eating stuff. And that human skill is really critical at the end to really think about, okay, now given the hypothesis of the different, sort of possibilities, what should I do? What can I trust? Maybe after I have 3 different analyses, right? To output, even for data analysis, and then you can evaluate them, with yourself But that's why with more recently we've been thinking like, how do you make the logic more explicit, right? And let's say okay, the ASA CCAM5 is really good. But I didn't have time to click into it, but basically the idea that we actually can ask AI to generate a comprehensive report on CCAM5 where it will show you based on this patient analysis why it is, for example, correlated with patient survival. What are the cell types that the CCAM5 have most highly enriched spectrum in myeloma patients? So a lot of these analysis needs to be part of so that you can have what we call explainable model for the AI system, so you can evaluate job, whether you trust it, whether you like it. So I think this is the second was like we still have a lot of work. If you think about AI as a helpful assistant, providing different possibilities and filter, and also make sure that the AI give you reasons why this is good or bad. Why he thinks that this analysis is very cool. Then I think human can really benefit from using this AI tools. I don't say we're just trusting something that we hallucinate, which definitely.
Interesting talk. I had a question when you spoke about the clonal populations.
Yes.
Is there a way to use lazy to track? Like, I'm wondering, like, the cells that do increase in fitness, are they just more plastic in nature, as in like they just have more chromatin accessible states, which is why they're able to adapt to drug treatment, and as a result, they are more resistant and they have a different prescription profile? Is the fact that they are plastic to begin with?
Yes, that's a weird question. So we recently started to rationale in UPenn, specifically for thinking about this idea of genetic stability versus genetic stability. So one thing is in sort of the supplement of one of the published work is we have to examine the genetic. We did actually target sequencing of panel, almost like an exome panel for the cancer cells to check if there's this genetic driver of this resistance, or it's an epigenetic process. So what we found is there doesn't seem to be indication of genetic mutation, like we call a secondary mutation that drives the expression. Rather, it is seems to be epigenetic driven, meaning that there is a subpopulation of cells that's in their own BRAF resistance, and they're all have a basic every mutation, right? But then within this population, there are some are epigenetic more prone to have transcriptional diversity or transcriptional plasticity, so that this subpopulation of cells are highly prone to be able to shift their expression, so that they can become more resistant, like upregulation on that, pathway. I mean, they have stayed. And what we think the reason why we like the ECH2 in the experiment we did is that ECH2, for example, is known to play a role in epigenetic modulation, methylation processes. So I think that to your question, like I think basically there are cells that are innately more epigenetically plastic, not genetically plastic, right? Not genetically more unstable. It's really epigenetic instability that are driving the different lineage expansion or non-expansion, at least in melanoma that we tested. We do think that this doesn't necessarily mean that every tumor cell or every resistant process have similar resistant mechanisms, but melanoma could be one of the most simple examples of this. We do think that we recently have started to play with pancreatic cancer models. But we think pancreatic cancer may also have that interesting aspect as well, based on our analysis of a pancreatic tumor cell. So I think maybe there are cell types that are more genetically stable. There are cells that are more genetically stable. And so for these genetically unstable cells or cancer types, the linear tracking will be super interesting about.
Very interesting. I have a question more so related to the RNA tool that you were discussing. So with RNA, I'm sure that when it comes to secondary structure predictions, there are sometimes an infinite number of secondary structures solely because RNA has other results going through it. So sometimes they pull in or pull. So I was wondering how what your method suggests is the most optimal way in narrowing down these structures, what necessarily is presented to the user in terms of how many structures you're presenting, and kind of how you're using RNA fold in conjunction with experimental data. Is that experimental data being provided by the user, or is it built upon a framework of pre-existing experimental data that your lab has built itself? And then on top of that, kind of, is this on a transcriptome-wide basis, or is it specific to the region or the sector that the person would be using and is it on a transcriptome-wide basis?
Yeah, great question. Maybe quickly two thoughts here. One, for a lot of structure prediction model, we actually need to input structure information from actually publish the starting structure for that module to actually work well. So basically, we can imagine the series model has been fine-tuned in the structure model with the structure predictions. And like I said, if you wanted to fit on have a family of RNAs, and you have fit on some example structure from, you fit them on my own lab, that actually would be super helpful in terms of improving the performance of the model prediction. That's one thing. And the second part is, I do think that we are just starting to think about a question like how protein and RNA action may be really needed to considering all the folding and stuff. And this is not explicitly modeled in the pre-printed version. So what we think is needed is to actually have a joint model where input can be RNA and protein, so that you can represent both. And that will be a more effective way. So we can think about the structure model, and then there's a sort of protein and virus structure model. So I think there needs to be almost like, out of the fold, right? You have the monomer and the multimer, which is sort of, and there's different modules. I think for a really powerful RNA model, it may need additional modules for different downstream tasks, so that it can really use the most helpful input and output for the task. So I think this is something we really need to do better, which is not yet in the preprint, which is more for the sequence test. So I think for the functional prediction that you mentioned, I think there needs to be multimodal input into the model that we should build like separate model for. Not going to be really. Thank you.
All right, in the interest of time, we're gonna have to move on.
Thank you.